Thank you, Luis. I hope you can hear me okay. I'm a bit funky and stressed. I might have an overtraining syndrome or red S, who knows. I'm really happy to be talking today. I passed the strenuous selection criteria of the scientific committee to be talking today. So it's really nice to be presenting this talk titled The Effect of Energy Deficit with Concomitant Exercise on Skeletal Muscle Phenotype Shift. And it's more like an integrative physiology outlook. So we're going to jump straight in about general sort of importance of skeletal muscle and the effect of energy deficit on it. So skeletal muscle represents about 40% of the body mass, and it's really a key tissue for both locomotion and regulation of whole body metabolism. What happens with skeletal muscle when there's an energy deficit? So typically it's considered this idea that the size of the muscle is reduced, so there's muscle loss, and consequently there might be a reduction in functional capacity and sort of suboptimal metabolic benefits compared to what would be maintaining skeletal muscle. And we've shown in the past that this is somewhat true by doing studies in trained individuals, looking at what happens with a short period of energy balance or energy deficit on skeletal muscle protein synthesis. So we've done like acute measurements of myofibrillar protein synthesis, so the contractile portion of the muscle after a period of energy balance or an energy deficit of a reduction of approximately 30% of energy. So in these studies, what we've shown is that we have a normal physiological value of fractional synthetic rate of muscle protein synthesis at rest. But then after 5 days, only 5 days of energy deficit, this reduces the myofibrillar protein synthesis. What happens when we acutely include a resistance exercise bout? This sort of brings the level of muscle protein synthesis back to normal. And then if we add protein intake after the bout of exercise, we see this going up, and it goes up further with more protein intake. So we know from these and several other studies that to maintain fat-free mass, resistance exercise and increased protein intake is a good intervention. So we know that the size of the muscle can be maintained, but it's very interesting that there's been very little investigation of what happens with the actual sort of phenotype of the muscle, and what happens with the physical capacity of this skeletal muscle. And now we want to move more onto not so much resistance exercise, but more aerobic type of exercise, and a series of studies that I think they're super interesting. This one is a study in mice, where they had like just a small group of mice for 8 weeks in 4 different interventions. So a control intervention, an alternate day feeding intervention, which of course would be very different for what is in humans, having in mice. So the impact would be a lot higher, I suppose, on having them exercising or having them exercising with a alternate day feeding. So what they see is that on average, this is the weekly food consumption for the mice. On average, per week on the alternate day feeding protocols, they eat less, but they maintain body weight in all the groups. There's no differences in body weight in the groups. There's lower energy expenditure in the fasting days, in the fasting groups. Interestingly, when they look at the skeletal muscle, there's an increase in markers of mitochondrial biogenesis in the exercise and in the exercise alternate day feeding that is higher than in the exercise groups and in the alternate day feeding groups. So when we look at different markers, so that's the green lines there, when we look at these markers of mitochondrial biogenesis, they are enhanced in all the groups that have both exercise and alternate day feeding. Interestingly, when they did with these mice test to fatigue, they see an increased performance in these mice. So showing that potentially this energetic stress is improving the capacity for physical performance in these mice. But we don't know if there's an effect of the energy balance of the alternate day feeding here. It's because it's a specific model. Maybe if we move on to see what happens in skeletal muscle in a model that is closer, let's say, to humans, when we look at these studies in rhesus monkeys, in macaques, where they have a very prolonged period of energy deficit. So this is the analysis of skeletal muscle of like older macaques that have been exposed for 18 years to either like a controlled diet or an energy deficit diet, caloric restriction of 30%. What we see when we compare like one group versus the other is that at this point in time, the monkeys with the caloric restriction, they show the same 24-hour exercise energy expenditure. They show lower sleeping metabolic rate. They are lighter, leaner, more active, and they have a lower metabolic cost of movement. So let's look at what their muscles look like. So the caloric restricted group, they maintain the amount of contractile area in the muscle, so this tends to decline in the control group. So the caloric restriction group, they don't lose as much muscle contractile area as they age, it seems, and the caloric restriction muscle is like a more youthful muscle. It looks like a muscle of a young monkey. When we look at the oxidative or markers of oxidative capacity in the muscle, we see an increase in cytochrome C oxidase, which is a very important mitochondrial enzyme, and this increase seems to be specific for type 1 fibers. Interestingly, and also adding to this idea of change of the phenotype, the lipid droplets, the amount of lipid droplets is increased and the size of the lipid droplets is reduced. But interestingly, in this group, there was, or in this study, what I say here is there was an increase in the physical activity as well in the group that has caloric restriction. So we don't really know if there's effect of caloric restriction. So this is to follow up on that. This is a very interesting study using overweight individuals, where they had individuals exposed to 6 months either of a controlled diet, a caloric restriction diet of 25%, or a caloric restriction diet that has the same amount of caloric restriction but inflicted both through dietary control, so reduced energy and increased exercise expenditure. So, as you would expect, if the experiment is well controlled, the weight is maintained stable throughout 6 months and decrease about 10% in both groups. So, looking at the biopsies, skeletal muscle biopsies of these individuals, what they show is that in the caloric restriction and caloric restriction plus exercise group, there's an increase in sort of markers of mitochondrial biogenesis, just as mitochondrial DNA, TFAM, PPAR, gamma, but there's no differences in enzyme activities. So this is data that, it shows us that I think we have very limited understanding of the effect of energy deficit on muscle phenotype and the effect potentially on mitochondrial biogenesis, if there's any. And this is also very poorly understood in people who have like high levels of physical activity or like athletes. And moving on to a little bit more of this population and to keep framing what the data that I'm going to present in a minute, is that we know that athletes with high exercise energy expenditure or reduced relative energy intake, they seem to have a couple of issues, a couple of problems, potentially, which is the affection typically of the hypothalamic pituitary gonadal axis that shows in women to reduce estrogen, to increase the amount of menstrual disturbances and result potentially in secondary amenorrhea, and in men decreasing testosterone, and we've shown before the data of Professor Jose Calbet showing clearly effect of energy deficit on testosterone, and there's also seems to affect sperm quality and quantity. Also, when we look at bone metabolism, the relative energy deficiency decreases bone formation, increases bone resorption, decreases bone mineral density, and there's an increased incidence of stress fractures. So it seems like, this is a little bit what we were talking about before. It's all this drama about how this thing is like all bad when potentially being done chronically, which it is a problem, but we don't really know what's happening with skeletal muscle. And we see that top athletes, like Kenyan athletes, before periods of or before competitions, are in periods of energy deficit, and they're performing at their best. They're winning their races. They're doing the best in the competition. So there's a bit of a paradox where this energy deficit might be having a negative effect on some things and positive effects on others. And following up on what Eimear just mentioned before, there's probably this idea of trade-offs of maybe rechanneling of energy. And this is a figure taken by this review from Zira and Harshman from 2001, where it shows if you look at figure C, where there's this idea that you have this number of 30. It's like aleatory numbers, the arbitrary number doesn't really mean anything for this case, but you have the R and the S, which are like the reproduction, and somas is like the maintenance of reproduction or the maintenance of the functions of the body. So in number C, it's just like equal distribution to both of them, but different species have potentially different energy allocation models, and some of them maybe maintain more the reproduction, others maintain soma. But the idea here being that when there is limited energy, there might be that there's some sort of hierarchy and priorities where energy is allocated depending on this importance for immediate survival. So bringing this to sort of a bit of a funky model that I've kind of put together with. This is just to share the idea more than a thought through and final model, is that when there is adequate energy, this energy is distributed as needed for different systems. So if we think about domains, some of domains can be actual, some of domains can be sort of virtual domains like growth, somatic maintenance, reproduction, locomotion, and physical capacity, then this, when the energy is enough, is distributed happily in an equitative way to all the system. But when there's less energy, potentially the systems that are getting more energy are the ones that are immediately important for survival. So the system takes energy off what is not important to stay alive, but more important puts the energy for what's more important to stay alive. And sort of in this little bit of a framework is that I'm going to present this data from this preliminary data that we have from my student Harry Taylor has been doing all the work here. So I feel like slightly ashamed to be presenting all the hard work that he's been doing. So thank you so much, Harry. And in this study, we had like 10 physically active young males that we put them through the following protocol. So we had them in our hands sort of thing for like 15 days. Not that we have them in the lab for the 15 days, but we had like 3 5-day periods. The first one was like a baseline assessment where we look at what they were eating, how much they were exercising. There were people that were exercising regularly about 2 or 3 times a week. And then we put them in a period of energy balance for 5 days where we were controlling the diet, we were controlling the exercise, that we were giving them the food, they were coming in the lab, we were controlling how much energy expenditure they have in the lab, and then we put them in this period of energy deficit or low energy availability, whichever way you want to put it. During this period, we used deuterated water as a tracer to look at what's happening in the myofibrillar or in the muscle protein synthesis, and it's an important data that I'm going to go through today. And through this period, we also looked at different points, body composition with DEXA, measured RMR. We took blood samples at different times. We took muscle biopsies at 4 different times. And with all this sampling, we look at different parameters, so more general sort of body composition, resting metabolic rate, and exercise metabolism data that I'm going to present today. When we zoom in a little bit more into the blood, we're going to present some data from testosterone and markers of bone metabolism. And then we're going to look in the muscle, the proteomics data that we've been working with Jotin, who's here in the audience now. We don't yet have the glycogen data and histochemistry, but this will come soon. So most of it is preliminary, but I hope it's something that is going to give you some information about what we're doing. In terms of exercise and dietary interventions, to give a little bit more detail, so we have people exercising on a bike during indirect calorimetry, so measuring the respiratory gases and calculating the energy expenditure through that. And they were exercising 3 out of 5 days of the intervention. The exercise was the same in both interventions. We targeted a specific amount of net exercise energy expenditure of 15 kilocalories per kilo of fat-free mass, so approximately 1,000 calories per session at an intensity that for these individuals was not particularly difficult, so around 63% of VO2 max, which is an intensity that for most people require quite a bit of muscle glycogen, but not an awful lot. And it takes about 90 minutes to complete on average for these participants. In terms of the diet, it's not a Michelin restaurant sort of meal, but it was very effective in terms of us being able to control the food that they were having. So all the food was provided. It's like you have this much of this much of that. They have check boxes, so everything is very, very tightly controlled. And when it comes to the actual energy, so the energy relative to fat-free mass was 54 versus 19 kilocalories per kilo of fat-free mass, respectively in each group. Absolute amounts, about 3,500 calories versus 1,200 calories. And what we did is we just made it smaller, everything. We didn't change the proportion of contribution of each of the macronutrients. So going straight into the results now. So we look at the body composition. As you would expect in the energy balance group, there was no net change of weight, fat-free mass or fat mass, whereas in the body composition group, there was about 3 kilos of weight loss in that 5 days. The majority of it was fat-free mass and was about 2 kilos about of fat-free mass and a kilo of fat mass as measured by DEXA. The proportion of body fat didn't change. When we look at the resting metabolic rate, we'll see the resting metabolic rate was maintained, so not significant changes. But when we look at the RER, that gives us an idea of the contribution of the substrate during the resting periods. When we look at here at the day 6 is the end of the energy balance period. There's an increase in RER indicating an increase in oxidation of carbohydrates by the end of that period, and by the end of on the day 11, by the end of the energy deficit period, is a decrease in RER, showing that there's more of a contribution of fat to resting metabolism. When we look at exercise metabolism, we see that during the energy balance periods, something similar happens. So the fat oxidation during exercise decreases, and during the energy deficit period, the contribution of fat increases as days go by. And we had a sort of a specular image of this when we look at the carbohydrate oxidation. When we look at testosterone, bone resorption and bone formation, testosterone first, as sort of we expected by the end of the energy deficit intervention, there's a decrease in testosterone. When we look at bone resorption, we were really not expecting an increase of bone resorption in the energy balance period. We're kind of scratching our heads around this, but there was a further increase in bone resorption in the energy deficit period. And we look at bone formation. We see a very clear effect of energy deficit in markers of bone formation straight after the induction of the energy deficit or the infliction of the energy deficit. There's a reduced bone formation. So now we're going to go into the meat of the study, no pun intended. And this is the skeletal muscle proteomics. So we're going to look at a little bit at the protein abundance and turnover rate data. So when it comes to the abundance data, we could detect changes in, or we could detect 2,400 proteins, 1,550 proteins in the soluble fraction, and 454 proteins from the myofibrillar fraction. And when we look at sort of turnover rate, we have 562 proteins from the soluble fraction and 114 proteins from the myofibrillar fraction. So this is work in progress. This is a lot of data that I'm sort of still finding my way around how to analyze it. Jordan is being very, very helpful. So this is preliminary data that I'm presenting now, but I just want to share a little bit of information about the sort of more general turnover of proteins and the soluble fraction data. There's a lot more data that we've generated that is we need more time to understand what's going on. But for now, I can share this, which I think is, for my liking, is really cool. Like, I really like this. So in terms of like this graph looks a bit funky. This must have a Redis, Neil. So the myofibrillar fraction data, we see no differences in sort of the global myofibrillar fraction synthetic rate, same with the soluble fraction, and same with the mitochondrial fraction. So when we look at, on average, what's happening with like the fractional synthetic rate, so despite the severe energy deficit, there is no changes after these 5 days in the turnover rate or in the protein synthesis rate. So this figure doesn't look the way it should. I don't know what happened when there's been some sort of issue with the transfer of data from my computer to this computer. But when we look at the skeletal muscle proteomics, when we look at the proteins abundance data, and we look at the soluble fraction, we could detect differences in 131 proteins between the energy balance and the energy deficit period, that when they are adjusted to a 33% false discovery rate, there are 100 proteins that show to be different. When we look at like the enrichment of cake pathways, these are pathways that look at a relationship between sort of metabolic pathways between proteins, and we look at the top 5. These pathways are related to, or the proteins are related to metabolic pathways, carbon metabolism, fatty acid regulation, fatty acid metabolism, and citrate cycle, the TCA cycle, Krebs cycle. When we look at the interactions, there's a high amount of interactions with this protein, indicating that they are like interrelated in terms of like how they interact in the metabolic pathways. When we look at what these proteins are related to in relation to cellular components, biological processes, and molecular function, we see that the cellular component that they contribute to are mainly mitochondria, so mitochondrial matrix, mitochondrial membrane, and mitochondrial inner membrane. When we look at the biological processes, are processes of oxidation and reduction, so, and TCA cycle in the mitochondria, most of them. And then we look at the molecular function, is like the oxidoreductase activity, as you would expect when it's related to mitochondrial function. When we focus a little bit on the mitochondrial metabolism, and we look at the top 3 clusters, so how these proteins are interrelated. So we have cluster 1 of proteins, that's sort of a list of proteins, I don't expect you to read it. It's mainly related to fatty, so beta oxidation. And then the second cluster is the mitochondrial protein trafficking and quality control, and the cluster 3 is the TCA and respiratory chain. So I've just thrown a lot of data at you now and trying to summarize it a little bit in one figure. So here what we saw is like the effect of 5 days of energy deficit with concomitant exercise on males. And what we see for now is that with the decrease of energy, or with increasing energy deficit, we see a decrease in body weight, decrease in fat mass, in fat-free mass, no changes in resting metabolic rate, increase in fat oxidation during exercise, and we see a decrease in testosterone and potentially a downregulation of bone metabolism. But when we focus into the skeletal muscle, it seems that there's signs of it being like an increase in mitochondrial biogenesis, and this is mainly affecting apparently the beta oxidation, the TCA cycle, and the electron transport chain sort of within the mitochondria. So that brings us to the conclusion. So first conclusion is data seem to be congruent with the idea that limited energy available is channeled towards skeletal muscle maintenance if there's exercise, and the muscle turning more like oxidative. It might be that some systems or domains may be downregulated, are maintained, and others upregulated. Energy deficit might promote enhancement of mitochondrial biogenesis in muscle. We definitely need to investigate what happens when there's no exercise or contractile stimulus present and what happens in skeletal muscle. And it would be very interesting to know exactly what is driving this. Is there like local factors to the muscle? Is it sort of more like humoral factors, circulating factors, and probably a combination of both? So that's all. I just want to thank the wonderful people that I have around that allows us, to do the research that we do day to day, our funding bodies, particularly the Alliance for Potato Research and Education, which allows us to fund this study that I just presented data on. And yeah, thanks everyone for listening.